Life. Welcome.
Good afternoon, everyone. Thank you very much for the introduction and for having me here. It's actually my first time here in China, so I'm super excited being here. So I changed the title a tiny bit to make it a bit more general, but I will talk also at the end about the origin of life stuff. So I changed it more towards AI augmented search of the emergence of self-organized living structures. And a big part of the talk will be about work done in collaboration with a synthetic biologist, trying to develop robust Turing methods and Turing patterns in microbial communities on the synthetic biology side, and us on the theory side and the AI side, and mathematical modeling as well. And then, now towards the end, I will talk about how this can be used for the origin of life, potentially. It's more speculative. So as theorists in particular, we think a lot about how to understand patterns in nature, to decipher these. Here on the left side, you see a nice little image from nature. There are lots of different patterns in this one image already. So you have the bird flock. This, one could say, is a relatively simple system because we can probably get away with pairwise interactions. We probably can neglect, details of the bird, what it had for breakfast or so. Then, of course, at the separate level, we have these beautiful stripes, a classical stripe pattern. That is quite hard to understand at this adult animal level, but if you go back to embryogenesis, then these pre-patterns have been developed very early on, when physical mechanisms or chemical mechanisms like diffusion and so on were very effective. Then we can zoom in, of course, and go towards microbial communities. And here we have hopefully a better chance because here we understand a lot of the ingredients extremely well. Of course, the physical mechanisms we understand well. There's diffusion, various chemical reactions in a cell. We understand cell growth extremely well. There's, of course, also chemical gradients towards the rim of the biofilm. Certain things can be controlled, let's say, in synthetic biology, others don't. In terms of mechanisms, we have lots of things to choose from: phase transitions, chemical gradients, Turing patterns, and that's what I will talk about. Here's a classic paper by Alan Turing from 1952. So he talked about two diffusing morphogens or chemicals which can form patterns. So they chemically react with one another, but they diffuse. The reactions themselves lead to stability, a homogeneous steady state. But when you turn on diffusion, counterintuitively, there's a pattern forming. Normally, you would imagine diffusion smears out molecules and it makes it more robust, but here you have the opposite. And he also talks already about biological applications and the difficulty of working in multidisciplinary sciences. Here's a little Turing mechanism in a nutshell. So it's different from phase transition in classical physics. First of all, it's non-equilibrium. It's a finite system. We have birth and death of molecules. Nothing is conserved. So we generally talk about bifurcations. Then there's a little model underneath. U is the activator and V is the inhibitor. There are chemical reactions involved, but also diffusion. And then you see these little diagrams normally with the arrows and so on, which illustrate it. On the left bottom part, you see without diffusion. In the UV diagram of concentrations, you have a stable steady state, which leads to the homogeneous profile. And this is stable with respect to perturbations. It would go back to the stable steady state. On the right here, you see when you turn on diffusion, a little instability is basically kicking the system out of the equilibrium, and you can form a wave number dependent pattern here, which is illustrated. So Turing already talked about these 6 different outcomes of these very simple models. There are oscillations, traveling waves as well, depending on the boundary conditions. Of course, here I'm mainly interested in the stationary pattern on the right, Turing 1. And so generally, you look at linear stability around a steady state, what happens to small perturbations. You look at the eigenvalues, in this case 2, as a function of this wave number, and then when it becomes positive, you can get an instability at a certain wavelength. There are some issues with Turing, so it has been essentially less popular than a later model, the so-called French flag model, because there's a nice morphogen gradient and it explains the positional information in a gradient that applies to Beacoid and Drosophila. There's also somewhat limited relevance in biology, but there are some good examples. So the digit formation in mice, the finger formation. It can explain mutants and is quite predictive, but it's not a mechanistic model. It's just a two-component model, super simplified. And on the right side, you see another nice example, the zebrafish skin pigmentation, where the model is also quite predictive, but it's based on randomly moving cells. So because of the simplicity of these models, the robustness of the models with respect to parameter changes is not guaranteed. And this is a major issue in terms of connecting to biology. So this is illustrated here, this robustness problem. So this was work by a collaborator of mine at Imperial College. So they scan through all potential 2- and 3-node networks, basically wiring them up in all different ways. So 3-node means there's one non-diffusing mode. And the thing is now is that most of these can produce Turing patterns, but only in a tiny fraction of parameter space, so illustrating the issue of fragility, which is a problem in biology, especially if you're trying to develop an embryo. So here's this quote was already earlier. So this is sort of the mantra also of synthetic biology. Feynman's quote about what I cannot create, I do not understand. So this is sort of the ideal approach here, because then we can build up complexity and understand where do you get robustness from. So this is basically the synthetic biology by my collaborators. So they took one of the most robust 3-node networks on the left and built it in every cell with plasmids. Every node is now replaced by 2 genes to separate out activation and inhibition. There are also certain knobs you can turn externally, like ATC and IPGT, where you can externally manipulate the pathway. There are small diffusing molecules as well, PC and CIO, which is quorum sensing diffusible molecules. That's essentially how the cells can interact with one another. This is work by my former PhD student, Martina. So she's then developed a mathematical model, 6 equations in line with the pathway. And you see there are sort of Hill functions on the right, which describe the nonlinear effects in gene regulation. So essentially, what you can see on the left bottom, you see a cellular automata simulation. So wherever you have cells growing, these equations are solved, and outside just degradation and diffusion. So here's the good news is experimentally you find all kinds of beautiful patterns. The problem is they're all done under very similar conditions. So meaning there's already the sensitivity to conditions you can't control very well. On the bottom you see the model. It can also describe these patterns quite nicely, but this is based on a global parameter search and then hand-picking the ones which look nice. So it's not particularly scientific and systematic. However, we can learn something from this theory. So with linear stability analysis, you can see again that Turing on the left is very fragile, or it doesn't occur very often, but oscillations happen much more often. You also learn that you have to be careful matching your reactions so you don't saturate them, so you essentially eliminate certain links in your node. And you also add ATC to make it more robust. Of course, one has to also keep in mind that linear stability analysis is only about the onset of pattern formation, and ultimately numerical simulation can lead to different outcomes. That's because of multi-stability. How to deal now with these big models with these 20 or 30 parameters? So first of all, this is sort of illustrated here. And that reminds you of the Van Neumann elephant, you know. When a model is complicated enough, you can essentially fit anything with it. So first we non-dimensionalize to get rid of some parameters. And it also eliminates concentrations because we have fluorescent intensities. And certain things we can also measure by, for instance, diffusion. Then we don't fit directly the patterns. We look at dose response curves in solution, in suspension cells, and then fit submodels to these with less parameters. And then essentially we look at the uncertain intervals of our fitted parameters, and then we explore these high-dimensional data clouds and see if there are any Turing patterns which look at like our experiments. So here's some results from a paper we published last year. So the patterns are more reproducible now, in particular with high ATC, which is good news. You see also regular spaced peaks at the top the green ones. So there's some definition of a wavelength appearing, equidistant peaks. You also see antiphase between red and green, meaning this is a prediction from the model. And there's no scaling with system size because, yeah, the bigger the colony, the more rings you essentially produce. And here's a time lapse and a simulation for illustration. So ultimately, of course, we wanted to fit a pattern, yeah? So that was the idea. So here we explored this, or Antonio, a PhD student, but in the end, we didn't manage to go from a pattern directly to the parameters given a model. Also, we would like to solve this inverse problem. So here we use numerical data. So basically, we're trying to identify when we have a good fit, the data cloud around it and how big these Turing islands are in parameter space. So that would be a signature of robustness. So we used very basic initially, we used very basic least squares fitting, where you maximize the likelihood of observing the data given some parameters. Super simple stuff. You see the Gaussian likelihood function. Originally, it was dismissed by other people because there are some potentially multiple solutions, but we don't care about it as long as we find some. And that was quite instructive. The main workhorse was, however, the physics-informed neural networks, the pins. And this is sort of illustrated here, if you're not familiar with it. So for pixels X and Y, you'd basically have a deep neural network to produce a pattern like the experiment or the numerical data. And then this is based on the universal approximation theorem. And then you can also do automatic differentiation. You get the differentials. And then you can also have in your loss function the PDE loss, the partial differential equation, has to be solved as well. So that's a cool thing because then you have 10,000 of parameters on the left, and then getting 30 or so parameters more for your model is not a lot of overhead. So there's a lot of good scaling. So here it shows basically least squares compared with pins. The idea is to take an image on the left from numerics and add noise, infer the parameters, and see the forward solved problem if it produces a pattern like the original one, which is the top row. So in the second row, we add noise. You can't see it because it's less than 1% in the second row. Here, second row, the noise is hard to see. It's less than 1%. And then the patterns resolved from here with the parameters inferred don't work anymore. So after 0.5% of noise, it breaks down, while here with the pins, you can add much more noise, up to 20%, 15%, and then it breaks down. But it's a bit more hit and miss. Sometimes it also fails at lower noise levels. So we can compare as performance measures the quality of the parameters, how they match the original ones used on the left for the original image. And we can also compare the patterns, but not pixel by pixel wise, but sort of based on Fourier modes on the spectrum, essentially on the wavelength. And this is shown here. So for 3 different models, you see least squares gets worse and worse in terms of parameters for more noise, but in terms of the pattern, the quality jumps, so it gets worse in steps because the spots are starting to move around and the pattern shifts. With the pins, they're much less accurate, and shown down here, but then it breaks down at much higher noise levels, so there's more resilience. We also explored the least squares more, so you can essentially crop images without noise, crop little pieces out, and you always can recover the full pattern, which is remarkable. But then we realized, you only need enough pixels to solve for the parameters. When you add noise, we sampled randomly, and for more and more sampled pixels, we get the no noise limit, which is just numerical imprecision down here. So what I said before, it breaks down at 0.5% of noise that would be rescued when you have larger images or more data. So we did apply it to data, but to chemical patterns which are well established and less noisy. So the problem with the biological patterns is they're very noisy. So these patterns are well established in this chemical system, and there's also a nice model. So we wanted to explore the inverse problem for the experimental data and the numerical data from the model. The problem is, on the left, this kind of data doesn't necessarily tell us if it's an activator or it's the inhibitor we're looking at, and so there are additional parameters to convert. But nevertheless, it works well for the numerics down here, and for the experiments, we recover spots, but we think that there are spots anyway, or it's a transition of spots. more recently then, with this talented undergraduate at Zichen, we also explored the problem of model recovery. So basically discovering the whole model. And I can't go into too much detail now, but here we use recurrent neural networks, so the dynamic version of neural networks. And the idea is now, that we try to train the network, but we formulate the network as a chemical reaction network, ultimately as gene regulatory networks. And when you then fit this model, or you can discover your gene regulatory network directly. So the deep neural network is the gene regulatory network. So that's the idea here. So that's sort of illustrated up here. So recurrent neural network looks like that. So there's some hidden states and so on determined by previous states. Here we try to, in this toy model, have a protein which is slow dynamics with a nonlinear activation function and the fast dynamics of the activation, which is like a promoter, let's say. And at quasi-steady state, this function here from the fast species approximates very nicely a ReLU activation function from deep learning. So the idea is now to scale this up and to basically build neural networks directly with simple gene regulatory networks. So this is illustrated here. So on one hand, on the left, you need quasi-steady state for the fast species to get the activation functions to use theorems from deep learning. On the other hand, you have to show with perturbation theory that the solution is also recovered for finite reaction rates, because in biology things are finite. So these two ingredients, and that was worked out by collaborators, is super important. Here's a little toy model. So we extended this to spatial patterns like Turing again. And we see on the bottom left, or on the bottom here, that the recurrent neural networks can produce the original one on the left. There's a little blurb here of errors, mean squared error, so the dynamics can be sometimes off. Here we see for different patterns the dynamics are completely off. We don't fully understand yet, but the final pattern nevertheless works very well. I don't know, my mouse disappeared. So the final pattern works very well. Okay, and so now I'm switching now to the more speculative topic, how AI could also be used to now reinvestigate the origin of life problem, which is one of the fundamental problems which are motivating many people in various different disciplines, from synthetic biology to biological physics. So on one hand, so it's quite a nice little mystery. So the question is, how to basically build a protocell which is then subject to Darwinian evolution based on random mutations and natural selection from basic ingredients. So you want some chemical physical mechanism, some replicator, to start simple and ultimately allowing you to get to the biology where evolution can kick in. So that's the question, which there's no direct answer yet. So one classic answer is the so-called RNA world, where we have already the inheritance molecule from biology, but also the enzymatic function included in one. Then there are different scenarios, where it might have happened, maybe hydrothermal vents, who knows. And then on the biological physics side or computational side, there are nice developments in terms of stochastic thermodynamics, self-assembly, dissipative structures. That concept has been around. Of course, phase transitions and all kinds of phenomena. And last but not least, of course, since the 70s, there are also these ideas around the panspermia, for instance, by Francis Crick, who raised the possibility, that life could have, or at least organic molecules could have arrived on Earth by meteors. Yeah, and then the last one is terraforming, so that then sounds more like science fiction, basically, where it's more intended, basically, the seeding of life on Earth. Of course, that's very speculative. There are some factual constraints as well, not a lot, but super interesting. So here's a bit of a timeline. So on the left, you see in the young Earth 4.5 billion years ago, it was hit by huge meteors, which are called sterilizing effects. So it would have been very hard for life to start at this time because of these huge blasts, which created the moon and apparently also brought water to the planet. But the interesting thing is, there's a note here, Luca, so the last universal common ancestor, was predicted to have started or lived already 4.2 billion years ago, soon after the water was essentially appearing on Earth. This is remarkable, and this is based on a very recent study on deep phylogenetic profiling of hundreds of bacteria and archaeas. That's quite an interesting, puzzling study. From here on, complexification happened, ending with dinosaurs and humans. I'm basically showing this to bracket a time window of 500 million years where life most likely has originated. So here's sort of the idea of this very simple approach here. It's based on information theory, and I'm basically interested in the question, what is the minimal rate of information which has to be there to build a simple protocell? Yeah? So you have to have some, for a given error rate you tolerate. So that's the idea, some distortion it's called. So you have some sort of rate of information transfer, and for a given error here, you have to be at least as high as that or higher. If you have too little information appearing, then you can't make it. And what is also important, which is shown on the right here, there's a minimum of rate of information transfer because we want to end up with a cell which has an information complexity of a protocell, and that was made during some available time t. So that's the minimum amount basically we have to come up with. So on the left, which is shown here, it shows the entropy collapse of the combinatorial complexity of chemistry based on some distortion. That is basically what's possible, what's hypothetically possible. Then somewhere in the middle, there's some chemical physical mechanism we don't know anything about. Technically, it's this prefactor eta, and then this has to be larger or equal than what is on the right to match the observation. Also here, so what is the AI connection here? So the information of the protocell on the right, this is basically where AI can come in because of recent advances. So what is this information content of a protocell? So here we propose to use a Kolmogorov complexity. This is basically the simplest explanation of a phenomenon, or normally use a computer code for that. What is the simplest code to produce something? So for instance, if you have a very simple string of zeros and ones, repetitive, ones and zeros, this is very simple because you just say you want a 1 and a 0 8 times. If you have a random sequence underneath, that's already much harder and you can't compress it anymore. If you have an image like that here, you could say there's a lot of information in here and that must be very difficult. But if you know it's a Mandelbrot set, it's a simple equation, then you would realize it's a very small information content. So that's sort of the idea of how to estimate the information in a simple cell. And so in reality, then one would use recent developments in whole cell models, on one hand, which have been established for small cells, also E. coli, I think now, with huge numbers of parameters and lots of submodules and so on. And on the other hand, we have, of course, what we already mentioned, AlphaFold and programs like that, which are very realistic and can solve protein folding problems. So based on the estimate of the codes in gigabytes, let's say, we can get an upper limit of the information required to build certain constituents. So here basically that's everything in a nutshell. So here, this is the entropy of this prebiotic soup, if you like. This can be estimated from meteors or chemical combinatorial chemistry. Of course, it's order of magnitude estimates. The distortion, the error, is essentially in here the lifetime of organic molecules. Here I used a day for the lifetime, but it could be minutes, it could be also technically billions of years if you look at meteors. So there's a huge variance. The available time, I said already, it's 500 million years. And the information of the protocell can be subdivided in genome, structure, and dynamics. And here we can use AI models or whole cell models. And then you find effectively that what is hypothetically possible is very high. 100 bits per second, which is roughly 50 base pairs per second. Yeah? But it's very optimistic, yeah? So there's no mechanism behind it. And on the right side, what you need is a tiny amount, technically only 1 base pair per year, or 2 bits per year. So it's totally possible that life has emerged spontaneously on Earth. The main problem here is that the mechanism in the middle is missing. It's of course a big question mark. And second, even so you don't need a lot of information rate on the right to produce a protocell in the given time, this has to be done every year for 500 million years. So that is the tricky part. So if you add now some random walk in this whole thing, you know, this will also sort of drifting towards the protocell. if we would add some random walk into this process with some effective diffusion, then you can solve for some persistence time with that minimum speed we had before. And the persistence time is like 250 million years, so that's huge. So so there's not a lot of room for random maneuvering around. So in fact, if you now go down in the persistence time of the random walk by 2 orders of magnitude, you get down to 1 million years. That would be already then takes the age of the universe to build a protocell based on these speeds. So there's some challenge. Of course, there might also be completely other mechanisms. I didn't talk about now in detail, phase transitions from physics. That couldn't happen more spontaneously, but then this mechanism is not well described currently. Okay, so I come to the summary. So I talked in the first part about AI-augmented design of robust Turing patterns, in particular the synthetic biology side and the parameter inference based on pins mostly, and then also briefly about the model discovery based on recurrent neural networks, but using already the gene regulatory network as a deep learning network. And the second part was about the origin of life, ray distortion theory, and this Kolmogorov complexity based on AI and modern codes to produce structures in biology. And at the end, I wanted to acknowledge my collaborators on the Turing side, Mark Iserlan in the synthetic biology lab, and his postdoc Yuri and PhD student George, and my students Martina and Antonio, and a postdoc Ruspe, who also helped out in some funding agencies. So yeah, I think I'll stop here, and if you have any questions, let me know.
Thank you. Very refreshing talk, actually. I like it. So I'm going to bring you back to the first part of your talk. So I find it interesting that you're working in defining synthetic biological systems, right? And yet you rely on fitting the loss function based on some measurements or on some curves, right? So have you thought about actually fitting the dynamical properties of the system that you're trying to build? For example, the largest Lyapunov exponent, or by proxy, some properties of the Fourier transformation or evolution of your system.
So I said one part I missed. So you said the dynamics. And what was the first part before?
So I was wondering why are you not fitting the properties, like the dynamical properties of the system that you are building? Right, and this comes from chaos theory, for example, right? So the largest Lyapunov exponent of the system, or much easier, so you calculate the Fourier transformation, and then you say, I want to see these components, because this kind of releases you from the need to follow one certain evolution of your system that looks in a certain way, but actually.
Yeah, okay, so the question was, why don't we look more at the dynamical properties of the systems? That's a very good question, so we thought about this as well. So one motivation was the synthetic biology again. So in synthetic biology, growing a colony like that takes like days, and you don't get a lot of dynamics because you can't image it constantly because of phototoxicity. The thing is dying basically when you look at it too much. So basically you have to work with snapshots or very few snapshots. So we were always more biased towards understanding a snapshot and finding a model to describe this given pattern. But I completely agree. I mean, there's a lot of information encoded in the dynamics, and how these patterns evolve with time can tell you a lot about the mechanism. So it's one of the future things we want to do. Yeah, it was motivated by the synthetic biology, but there's otherwise no good reason to do it or not to do it. Thank you.
Thank you, Prof. Robert. Thanks for your nice talk. I have questions about whether you consider using the manifold learning to modeling the bird flock. Is that manifold learning is able to the UMAP? You can look model both exposing the both local and global structure. Is that not just compare with PIN or LS that you have developed? Yeah, just based on the metric-based method. Yeah, using the manifold learning to learn the patterns. Yeah.
Yeah. A good question. I mean, the bird flock, you mentioned the bird flock, yeah? So the bird flock I mentioned just as an example where people work on patterns. So I myself haven't worked on on bird flocks. I mean, manifolds, yeah, I haven't used them actually. So it's more like a clustering mechanism, right? So like autoencoders. So autoencoders we used before, finding low-dimensional representations. We used them, I think, for morphodynamics of cells to find sort of the shapes underlying the patterns of cellular shapes. But for a Z problem, we haven't used it. So of course one could, also one of my ideas was also to learn the neural networks based on numerical data, because we had a ton of it. So we had tons of different patterns, thousands of patterns from our complicated models. And then we could learn the structure of this pattern space. And this is a bit like maybe what you are asking. So that was also on our list to do, yeah. So that would be super interesting.
Yeah. Thank you.
Thank you.
Actually, I have a question. Because we use AI models to predict something that happened 1,000 years ago. It's very hard to get the ground truth to validate what we predict from the model. How do we use, or how can we convince that non-computational biologists to believe in what we predict from the models? Especially when we communicate with these biologists, they will say, you can predict anything, but how you can make sure that your prediction is reliable and trustworthy?
I mean, of course, yeah, it's a good question. Yeah, I mean, I don't know if I can answer it so well, but of course, we want to be consistent with the data we have. I mean, there's not a lot, but there's, of course, in terms of origin of life, fossil data, which I didn't mention now, 3.5 or almost 3.8 billion years ago. And of course, this one study I mentioned with Luca is actually a bit going in this direction. Yeah, it uses a lot of machine learning for the deep phylogenetic profiling. So there's already set aspect of deep learning or machine learning in it, but it's also quite, makes a big effort to be consistent in terms of all kinds of other knowledge we have. You know, when you discover certain components of a pathways which might have occurred in these protocells, then you can use information about signaling pathways to fill in certain gaps or to be consistent with whatever we know about early Earth. So of course it has to be in line with whatever we have, the information, and ideally it makes some prediction, right? Ideally it would tell us, you know, how to make some cool new organism in the lab, and yeah, this recipe would be really fantastic to have, yeah.
Thank you, Robert. Okay, coming up next will be 2 flash talks we selected from the poster submission. And the first one will be given by Dr. Deng Pan from Beijing Zhongguancun Academy. She will also present her work on virtual cell modeling. Let's welcome.
Good afternoon, everyone. I'm Pan from Beijing Zhongguancun Academy. For those of you who never heard of this academy, we are a new artificial intelligence academy established last year by Ministry of Education in China last year. And today I will talk about drug discovery with virtual cell modeling. So when we talk about AI in drug discovery, we usually think about the hit identification and lead optimization. We will focus on the disease-relevant targets, protein targets, and try to design and optimize therapeutic molecules that can bind to and thus inhibit the targets. So this is a very important step to work with. But if we look at the whole pipeline of drug discovery, we will find that the molecular discovery is only one step in the whole long pipeline. Actually, 90% of the drug candidates will fail in the end, and most of them doesn't happen at the drug discovery step. Most of the time and the money spent actually spans the whole pipeline from the target identification to the preclinical and the clinical phases. So this demands us, we need to develop a method that can develop the drugs at a more systematic level to improve the success rate of drug discovery and to fully unleash the power of AI in drug discovery. So leveraging the most recently progress in virtual cell, I think the speakers previously also introduced to this concept, we're trying to see whether we can use the artificial intelligence and the virtual cell modeling technique to improve the drug discovery pipeline. And we want to say that the virtual cell modeling can actually extend its power in the pipeline to both upstream and downstream to have a broader coverage on the drug discovery process. So I will give you a brief example and actually our progress on these directions. So the first is the target identification. So what does target identification mean? If we compare a diseased cell to a healthy cell, we may find that many of the genes are expressed differently between these two conditions. However, there are only a few of these genes actually the root of these changes. They drive all the changes of the other gene expressions. So target identification is to find the most upstream gene in this process so that we can target to revert the disease progress. So our goal now is to identify the driver genes in the disease progression process. And for this purpose, we have developed a method called CellNavi. It is a deep learning framework for driver gene prediction and just published on Nature Cell Biology this month. So basically, the definition is given a pair of cells, a pair of healthy and probably diseased cells. We want to predict the driver gene that is responsible for this conversion process. Like it can drive the cells from the one state to the other. Here's a very brief touch on the design of the model architecture. It's actually kind of the very common single cell foundation model approach with a self-supervised learning on the RNA-seq counts. But here we had 4 different designs. The first thing is, aside from the single cell RNA-seq data, we also create a cell type specific gene graph for each cell. So we believe that the graph structure is more natural to describing the gene relationship in a cell. And then to couple with this design, we introduce a so-called the gene graph attention layer to better leverage this graph structure in our input from our data. And next, we use a bottleneck architecture. So we force the model to learn more informative cell embeddings in the process. And we ask it to recover the cell expression from a very small cell embedding of probably over 20 or 500 dimensions. And lastly, we use a downsampling reconstruction task to help this self-supervised training process. And we can see that with this cell representation model, we can align the sequencing of the same cell of different sequencing depths. And also it helps to recover the developmental trajectories for the classic development process. Okay, so now our next step is to predict the driver genes that alter the cellular states. So here we fine-tuned our model with CRISPR perturbation data. It is a perfect supervised training data because it offers the causal relationship between the start and final state of cells as well as the driver genes. So by inputting the source cells and target cells to the model, and through the previous cell representation modeling, and here we build another driver gene predictor. I skip it here. We will have the ranking of the candidate genes that are likely to drive this transition process. So one thing is most of these CRISPR experiments are done in systems that are highly homogeneous, like cell lines or mostly immortalized cell lines. But we want to apply it to more clinical side, like in primary cells or patient tissues with varying conditions and higher heterogeneity. So this pre-training strategy allows us to transfer the knowledge from the CRISPR screening to more clinical side. And to demonstrate this generalization ability, we assessed CellNavi on 2 benchmark data sets. These are well-known data sets in single-cell perturbation studies, but we use it for reverse purposes. And to make sure that we can test the model's generalization ability, we make sure the train and the test are split by cell states instead of random split. And here, the red colors are the results for our model. And you can see that compared to the other available baselines, our model can greatly outperform them on all different kind of benchmark metrics. And for application, we tested it on three cases. The first is we try to identify the master regulators driving the T cell differentiation. And here it shows the differentiation of tier 0 cells into the IL-2 high cells, interferon gamma high cells, as well as Th2 cells. And our model can successfully distinguish these driver genes for different cellular states. And the second step, as I mentioned, we're trying this work on for the drug target identification. And in this neurodegenerative disease system, our model can discover the known drivers for this process, as well as nominating new candidate drivers for this process. And the last very interesting thing is we can apply this model to elucidate the mechanism of action for drugs. So here, one thing I want to point out is we didn't do any compound-related training at all. But by applying this model, we find that the 17 different HDAC inhibitors may show different downstream cellular program preference during their functioning, suggesting that these HDAC drugs may have different mechanisms in cells other than regulating the histones. Okay, so here's a very brief summary on the CellNavi. It is a deep learning framework designed for driver gene prediction, especially in drug discovery context. But during our study, we noticed there's a lot can be improved further, like we want to further explore the long-distance cellular transitions and to rebuild the transition trajectories to better support the cell therapy. Also, we also want to see how far we can push it, the generalization boundaries, so it can be applied to novel scenarios and novel genes. And I'd like to use the last 1 minute to introduce our recent work on designing compound using cellular models. It basically falls back to the paradigm of phenotypic drug discovery. So here, the definition is we have a pair of cells, diseased and healthy cells. We want to find a compound that can revert these status. There are many benefits about this method, like less cellular toxicity, but better activity. But people will criticize it for not knowing the target, and now we can use our previous method, CellNavi, to ID it. And this work is still ongoing, so I'm going to just show you very brief results here. In collaborating with Westlake University, who is doing the wet lab validation for us, we built a model that can generate candidate compounds directly from a pair of cells. And we tested it on 3 different out-of-distribution data sets. They have like unseen compounds or unseen conditions or genetic perturbations. Our model can greatly outperform other baseline methods as well. So I'm really excited to see the wet lab results coming back. Brief summary here. I'm going to stop here. I briefly show that how virtual cell modeling can bridge the molecular level perturbations to cellular phenotypes, thus can help with the drug discovery. I showed you our progress on target identification and the hit and lead development with cell models as well as the molecule models. And the last thing is clinical evaluation. The perturbation response prediction is a very hot topic nowadays. I think everyone have heard about it. And we also expect to have reach some progress on this topic later. Okay, this is all for my talk. Thank you.
Awesome. So I guess I missed the information how you integrate the heterogeneity information to the model for your predictions. Thank you.
Okay, thank you for the question. And sorry for being so fast. Like the time is very limited. You mean the heterogeneity in the samples? Oh, yeah. We don't need to explicitly model that. So basically, we trained on the CRISPR perturbation, and since we leveraged a universal cell representation model that can generalize to different cell types, so it by nature has some generalization ability to the system it has not seen. So if a heterogeneous system that is kind of similar to some immortalized cell lines with CRISPR perturbation data, then the model can transfer from this side to the other side.
All right, thank you.
Thank you, Dongpan.
Thank you.
Okay. Next talk will be presented by Chen Zeyu from Peking University, representing Ma Mingqian from Carnegie Mellon University. They will present their work on DNA language model. Let's welcome.
All right, thank you, everyone. It's my great pleasure to present our work here about a second version of hybrid DNA. My name is Ziyu Chen. I'm assistant professor at the School of Basic Medical Sciences, Peking University. And today is actually an updated version of hybrid DNA, which is an efficient DNA language model for genomic complexity reconstruction and context-specific interpretation generation. So why DNA is important? Why genome is important? Because there's actually a lot of applications for dissecting genome functions in both understanding biology as well as pharmaceutical. First part is actually pro-diagnostic values for understanding how genetic mutations that are affecting different phenotypes of different humans. And second, more importantly, we can basically use DNA or RNA as a basic language model and input into different cells and to learn whether gene therapies or cell therapies, they have better specificity and efficiency during these processes. So when we really understand DNA as a language, what are the core features for these DNAs? And it's basically comparing to other languages. First, genomic DNA is long, and of course, other DNAs are long as well. So we definitely need a long input and the screening systems. And the second is genomic DNA is both linear and nonlinear interactive. We know that there is a 3D genome. We know that the enhancer promoters kind of attaching to each other actually functions together. And third, that is actually really basic, but super important to understand DNA as a language is reverse complemented. So actually the original versions of hybrid DNA was published on arXiv early this year, and then basically mainly by Ma Mingqian, Liu Guoqing, Cao Chuan, Deng Pan, and at Qintao's group, now at Zhongguancun Academy. And then this model was a mixture of the transformer and the Mamba2 together, which both actually can get the long-term input embedding of all these DNA tokens, all these DNAs, and also single nucleotide token attention by the transformer layers. So basically, they mixed 7 Mamba layers with one transformer layers together to basically get balanced efficiency as well as the specificity for this model. And so we got a really an efficient model comparing to a lot of other transformer models which can be run in a small computer in general. And then we pre-trained this model by multiple species data set, including over 800 species and then 160 billion nucleotides. The 3 model size that people can choose to use in their different size of computing powers, like 300M, 3B, and 7Bs. And also, depending on the use case capacity, we can use 8K, 32K, and then 131Ks. So last but most important is can perform both genomic DNA dissection function and functional generation tasks. So on the current second version, one thing that we mainly put into an updating of this model is because we consider the non-linear interactive features of these next token prediction processes. We basically put the onco-embedding features of hybrid DNA into an RC. So we basically put the reverse complement DNA and recapitulate the onco-embedding processes. So this is actually a pseudo bidirectional decoder system rather than encoding system. And by posting embedding the long-range DNA classification, we can basically reconstruct the complex trajectories over multiple species. So what we did here is basically we take the testing random 8K from this testing set from different species and directly embedding into latent space. And now we do the dimension reduction and form these UMAC clusters. So we can see that there is clear distinguishment between bacteria, fungi, and the other later higher species. But what's more interesting that if we actually directly put a molecule, which is a single cell using pseudo-time reconstructing system into this embedding random 8K genomic DNA, we can reconstruct this pseudo-phylogenetic tree. Currently, we cannot call it actually an evolutionary tree yet because we are lacking of common ancestors, but at least we can dissect how complicated this DNA changing during this generation process by adding different species head. So next round, we want to really doing something that is potentially pharmaceutically valuable, which is designing the cis regulatory elements that can go beyond the evolutionary regulation limitations and get stronger context-specific responding features. Because just one brief introduction of the regulatory elements, we know that the gene expression require a lot of DNAs that is actually by the enhancer regions can bind to the core promoters and drive these gene expressions. And these cis regulatory elements, first, they can critical in controlling gene expression, and second is essential for tissue-specific functions. But current limitations, because natural CRE sometimes, because there is actually a limitation in evolution to control the gene not expressing too high, it's inefficient to precisely for therapeutic applications. But currently, if we want to put into a pathological conditions, which is never seen in evolution, then in that case, we can actually drive it further. So we want to do synthetic DNA sequences. And there are two things that once you are actually really facing a pharmaceutical use case, you want to actually to get it really explainable. So first, how to dive into generation processes and get a higher specificity and efficiency. We try to use reinforced learning. And second is how to interpret the generation process and correct the stochastic during this generation. We deconvoluted the reinforced learning processes. So the iteration reinforced learning process is quite simple. So basically, we actually use hybrid DNA generating process, generated sequences, and using a scoring model. We tried a lot of scoring models. The details can be on the posters. And score the sequence and refeed back to the hybrid DNA processes. By this reinforced learning processes, we actually outcompete the controls, the negatives, and the nationals, and also the malinos, which is now the SOTA systems in the generative art. So have the high score DNA, how it looks like, and then what's the DNA process really learns, and how much money will cost in this process. So first, to control how much money will cost, we use an iteration-based deconvolution to see what's the margin of this saturated computing. So the x-axis is basically how many iterations we go, and the y-axis are the motifs that we use as a vector to actually scoring all these generative sequences. So we can see that the two first cell lines, KFS2, SK, and SH, was saturated pretty fast, but the later one, HapG2, was actually saturated later. So we take the HapG2 as one example and see what's the next to see what's they really generated on different iteration layers. And we see that there is labeling different transcription factor motifs, including HENF1B, FOXA1, and NR5A2s, which these transcription factors are really interesting in these processes. And then we now dive into the layers. The first to the 15 layers of this iteration, the hybrid DNA model learned the mammalian regulatory elements contains GC sequences. And in the 50 to 100 layers, it learned most of the motif sequences, including the important one, FOXA1. And then the later ones is super interesting. They learned only one feature of this process is HENF that is gained, which if you correlated this HapG2, which is hepatocyte cell line, with the liver development, in generation process, our FOXA was earlier than HENF that is present. And in the liver development, the FOX is earlier than HENF. So we are planning to actually using a real liver developmental model and trying to say if there is anything correlating with the iteration during the generation and the real developmental processes. So next thing, if we can actually really generate the functional elements, we try 3 cell lines, and we see that the generation process have really high specificity during this process and in these 3 cell lines. And then we did white lab experiment. Here is one construct that we designed. We make sure that our inserted sequences did not interpret with the original vectors, and then we divided into 4 subgroups. And we one thing that is we are really lucky, at least in these HapG2 cell lines, we got super high specificities. The y-axis, the lower it goes, the higher of the generative enhancer intensity, and then the x-axis actually labels what actually sequences it was originally generated from, so we can HapG2 is really specific. When we dive into these ones, they really enrich in a lot of HapG2 specific enhancer motifs. And then once we actually get this out into single construct validations by flow cytometry, we can validate that both HapG2 original and HapG2 RC sequences are actually really high, significantly higher than the random. So for summary for this study, hybrid DNA is an efficient long-range DNA model with the long-range DNA dissection and the generation capacity. It enables recapitulating multiple species genomic complexity trajectory with a random 8K. We didn't watch any of the data. It's a random 8K DNA fragment input. And we can actually use iteration-based deconvolution to interpret the hybrid DNA generative process through reinforced learning layers. And generative DNA elements show strong activity in a context-specific manner in wet lab experiments. So thanks to everyone, particularly Ming Chen and Wan Juan for these studies. And we have actually 2 posters if you want to learn more details on this study. And questions?
Hi, my name is Tang Tao. So is the usage of member tool for handling large-scale genomic data?
Yes.
So has other structured state space model been tested, like the HYENA or?
Yeah, that's a really good point. Actually, before actually getting these hybrid DNA models, we use CNNs. We also use Luca Pinallo's group DNA diffusions. They all get pretty nice results, but we feel like hybrid DNA, because it has both member and transformer, it's pretty flexible, so it's actually really efficient. You can run it in a small computer like the 4090 plus 2, this kind of things. So it's a really light model, and you're using a 300 million, I think, token models, and you can actually get really context-specific enhancers.
By the way, may I ask why the architecture is so process the data with multiple layer member than one layer transformer and multiple member again? So what's the...
That's actually a really interesting question that
That question is a bit tricky, but so...
Yeah, it's quite tricky. So basically, this is originally from the original member hybrid DNA papers. They tested a lot of combinations. Their assumption is the DNA is super long. The genomic DNA is 3 billion long, and then there was actually multi-interactives. So we need to use member to actually just scan where's the DNA and embedding as long as possible. So this is actually much more cheaper than if you're embedding a super long DNA with transformer. And it's completely not economy. But also you needed actually transformer layer to dissect the single base tokens and see the function. So to basically be both broad and precise, we fuse the member and transformer decoders.
Yeah, absolutely, thanks.
Yeah, thank you, Zoe. Okay, I think now it's at the end of today, and that's all for the science part for today. Sorry, it's not at the end of today, because we will have our welcome reception dinner, which will come in 15 minutes, so we can have a short break. Thank you again for all the fantastic talks and also the discussions. I think it's really, really interesting, and we look forward to more interesting discussions tomorrow. Just a reminder, if you take the shuttle bus to Zhongshan Hotel, it will depart from here at 8 p.m. and it will come back to the conference hall tomorrow at 8 a.m. So just as a reminder for you, 就是去中山宾馆的这个摆渡车